30 seconds apart. And then the other one an hour later. So you can see our site is pretty clean. We have, on all 3 samples, we have 0 small wells fluorescing and 2 large wells fluorescing. And so then you take your MPN chart and you find large wells 2 and small wells 0. And so all 3 of these yesterday at Pier 40, in those moments that I took a sample, we got 20 most probable number of bacterial colonies.
Is this fluorescent?
Okay, so sometimes other bacteria will eat our reagent, and those might fluoresce other colors. That's a very good question. We are only looking for the fluorescence that is bright blue. So sometimes you do get a really bright yellow or a sort of greenish color, and that just means a different bacteria has eaten the reagent. So that's not what we're looking for.
What was the number you got again?
20. And actually, I have reagent or samples from last week. So if anyone would like to, we knew we were running late. So if you guys want to stay behind, you can ask us questions, and you can also, I can pull out more samples, and you guys can practice using the UV light and using the MPN charts, or you can come down onto the floating dock with us and practice taking a proper sample if you're interested.
How do you get the water into here? Do you just pour it in the bag?
Yeah, you sort of put your fist in and open the bag up really carefully, because also these are sterile as well, so you want to be really careful not to contaminate the inside of these. And you pour it in, and you have to sort of lay it flat and watch the water flatten out and fill up each of the cells. And in the beginning it is a little tricky, but really you want to be careful of the small wells, just because if those don't fill up, you sort of have to avoid your sample if one doesn't have any water in. The larger wells actually are much easier to tell. And then actually this big guy up at the top that catches whatever is left over actually counts as one of your large wells in the IDEX system.
And then you seal it with a hot sealer.
Oh yeah, and it's basically like a big laminator that you use to seal them, so that once they are in the quantitrays and they're in their wells, the water can't go in between any of the others. It's just trapped in its one well.
So how much does that cost?
Me and Tracy, I guess, have a little bit of different calculations. So Tracy said about $15. I've come out, like, as long as there's no like person time working in the lab or anything, it's about $17 per sample, I would say. And really, our
Is that with the kit?
the most expensive thing is our sterile water and the reagent. So obviously you have to buy the reagent, but if you could find a different way to get sterile water, it could be much cheaper.
Can't be just boiled. Boiled water doesn't make it
Technically, you
sterile.
probably could sort of boil water, but how would I actually know that my water is definitely sterile? I don't have the good enough testing equipment to really be able to say, like, this was definitely sterile. And we are not a state certified lab, and just started doing this because there was interest in our community, and sort of we saw this data gap that we wanted to fill, that voters felt that their sites weren't getting tested. So we really wanted to fill that gap for them. That's why we started.
Good for you. Touche.
Any other questions?
How does 20 rank?
20 is green, that's swimmable. Up to, actually I think the EPA's new guidelines have a little bit of different numbers, so I'm not positive, but up to 50 MPN was swimmable is what I should say as of last year. Up to 100 was in the yellow, so you could swim in it, but you shouldn't stay in it for too long. And if your testing is consistently getting a yellow status, there probably is a problem, maybe a small leak somewhere, and someone should check it. And then above 100 is red, and you should not really go swimming there at all. There's definitely something going on.
A little indirect question. When the discharge occurs at a location and the other occurs in that, do you have any information on how that changes over time based on that one event, either due to dilution or due to whether they multiply for some period of time in the environment?
We have talked with the Stevens Institute a little bit about that, but I'm not confident really saying anything too much. I know if it's really sunny days afterwards, like the UV rays, if they're up in the top of the water column, will kill off a lot of the bacteria. But there is a lot of mixing. If there's a lot of sediment in there, there's a lot of things for the bacteria to attach to and find food, and be able to reproduce and things like that. There's just a lot of things going on. And we are just hoping that we do this weekly every Thursday for 20 weeks of the year. We did this, and this is our second season. We're about to complete it. I think we have 3 more weeks left, so we're just hoping if we consistently test every year that patterns will start to emerge if we compare it with rain. And at Pier 40, I do take dissolved oxygen, turbidity, salinity, and temperature right when I take my sample as well. And then obviously there's lots of tidal data that you can find. So we're hoping that patterns will start to emerge, but we don't really know anything yet.
Is there an expert here?
At Stevens?
Yeah, that focuses on that question.
I just sort of call them up sometimes. I have a couple people that I've worked with, and Alan Bloomberg, I believe his name is, has spoken with me before. But we're really just in the beginning stages of that. Yes.
You know you take it every Thursday, test. Do you ever just out of curiosity take it after like a whole day of rain?
No, we usually don't do that. I will take some sporadically. I know after Sandy, everyone really wanted us to take samples, but we didn't have any power either. But really, it's just sort of me and Catherine and Fiona who run this program. We're a very small nonprofit, and I do disinfect the lab before I go in there and do anything in there. So there's really a lot more to think about than just going out and scooping up a sample and throwing it into a Quanta tray.
Dan, I'm one of the samplers, but it does work out that it does. There have been large rainstorms on the Wednesday night or the Thursday morning while we're sampling, and they always post it with the rainfall data from that day, because that's part of what we're trying to figure out.
All right.
I have a site that's a little bit worse, though not the worst.
Okay, cool. So Catherine has brought some more samples.
Sometimes,
Okay, good.
sometimes it will be completely blue. So that's a sample where the number of bacteria in our sample is off the charts, and we just have to say greater than
24,000.
Yeah, 24,000. So this one we can still count. Yeah, the number of colonies. So this is one that looks a little bit worse than the one from Pier 40.
So I guess if there are no more questions, I think if you guys want to split up, we do have a couple samples that I hadn't gotten rid of from last week. If you want to come see what using the MPN chart is like, but if anyone would like to come out and practice taking a sample too, we could do that as well.